inviting me. I would have really loved to be there in person with you. I would have enjoyed this a lot more. But hopefully still some use doing it like this. So I'm going to talk to you a little bit about some of the things we learned from a project where we wanted to look at evolution of E. coli in the wild. And when I say the wild, this is a picture of the wild. So this is on the shore of Lake Superior in Minnesota. There's a parking lot with a landing where you can go and let your boat, or you can go fishing. And the group of Mike Sadowski about 15 years ago went there and collected samples from the water, the watershed soil, and soil 1 meter inland of this there at the shore. And they did this within 1 month. They went 2-3 times, collected a whole set of E. coli's, and they send us 5 plates with strains. But today I'm going to focus on the results from 1 plate that we started with. So, and we got a lot of phenotype and genome data, but today I will not talk about the phenotype data. I will focus entirely on what we learned of genome data. So after we've sequenced and assembled these genomes, we made a core genome alignment. So the pieces of the genome that are shared and that occur once in each of these strains. And we made an alignment of this core genome together with those of a collection of reference strains from the database. And here I'm showing you the phylogeny that you get if you apply a maximum likelihood phylogeny reconstruction to that core genome sequence alignment. So in red are all our strains, and they're just named after the well of the 96 well plate they were in. In black are other reference strains. Green are some well-known lab strains. And I guess what the first thing that you should get from this is that we find in this one place, from this one place in one point in time, we find sort of the full diversity of E. coli genomes that is known. So we find strains of E. coli that go all over the known phylogroups and phylogeny of E. coli. And we even have here one outgroup of strains, so the length of this branch is actually shortened, which is really actually something like 6-7% diverge from all the other E. coli's, whereas the other E. coli's are sort of like a 1 to 3% within each other. All right, so one of the first questions is, so what is this core phylogeny represents? And as you all know, one of the miracles of biology is that every cell is the result of a cell division, which is the result of a cell division, which is the result of a cell division. And so it's all connected cell divisions all the way back to the early life. And so if DNA is transmitted only vertically through replication at cell division, then from the patterns of the DNA at the leaves, so of the strains that you actually sequenced, you should be able to reconstruct this clonal tree. That is the tree that reflects the cell divisions. However, as is being known for a very long time, and most of you probably also know that bacteria are able to also acquire DNA horizontally. They can take up chunks of DNA from their environment. A virus may package DNA from a host and transfer it into another cell, or you may even have exchange of DNA via direct contact between cells. And when the endpoints of these chunks of DNA that are being transferred have homologous endpoints in the chromosome of the cell that they enter into, then through homologous recombination, the DNA can get integrated into the host genome. And of course, when such horizontal exchange is happening, those parts of the genome that have been subject to this exchange may follow different phylogenies than the clonal phylogeny because they, come from different ancestors. So one of the questions we wanted to look at first is to what extent the phylogeny that we reconstructed these strains, what is the evidence that this is actually representing the clonal phylogeny? And so this is one of the main puzzles that we've been looking at in this project. And this puzzle is that if you take your whole genome alignment and you take any short segment from it, so let's say a block of 3 kilobase long, and you build a tree from this block, then you find that for every block you pick, the tree you get is statistically significantly different from the block that you get if you take the whole, from the genome, from the phylogeny that you get if you take the whole alignment. In addition, all these 3 KB blocks all reject each other's phylogeny. And in this picture on the left here, I'm showing you the core tree, that is the phylogeny reconstructed from the entire core genome, which is about 3 megabases long. And in the colors, I'm showing you what fraction of 3 KB blocks have each of the branches that occur in this tree. And so as you can see, actually, most branches in this tree are poorly supported by the phylogenies that you built from individual blocks. However, if you take any large number of blocks, right? So you sample randomly half of the blocks in your genome and you build a tree from those trees are all very similar to the tree that you get from the whole alignment. Okay, so as you take more blocks, you start converging to some sort of clear phylogeny. All right, so an obvious interpretation is to say, well, this phylogeny then probably represents the clonal phylogeny. This phylogeny that you converge to as you take more and more blocks. And one way to think about it is that this recombination, this horizontal exchange of DNA is essentially just introducing a noise onto this clonal background. And if I just take a small block, then this small block, the noise may be big enough to distort the signal so that I will get a different tree. But if I take many blocks, then this noise from this recombination will start to average out and I will converge to the clonal phylogeny. So we decided to ask, is this really the correct interpretation? And are there ways that we can confirm that this is the correct interpretation? So I want to tell you a little bit about some of the sort of methods that we decided to use to do this. So one method that we looked at is to look at pairs of strains. And what we noticed, and I want to say this has been noticed before, there's a nice paper by Dixit and Maslov that has looked at very similar patterns, is that if you take an alignment of 2 very closely related strains, so this is a pair of strains, which is the 5th percentile closest pair of all the pairs in our data. So their divergence is 4 times 10 to the minus 4. If you now make an alignment of the 2 and go with a sliding window over this alignment and ask what is the number of SNPs per, let's say, 1 kilobase block, then you see that for most of this alignment, this number of SNPs is very low. And then there are here 2 regions where suddenly the number of SNPs is much higher. And the number of SNPs here, you see this is around 20 SNPs in a KB, which is sort of the typical distance that random pairs of strains have. So these 2 strains are very close almost everywhere. And then there are these short regions, which is of 10, 20, 30, up to 50 KB or something. And in those regions, the SNP rate is something like what you would have with a random E. coli. And so the obvious interpretation is these regions here were actually recombined, and one of the 2 strains took a piece of DNA from another E. coli. Now, as you go to strains that have larger divergence, so here I went to a divergence which is like 5-fold higher. This is the 7th percentile, so it's still a very close pair. You see that now the number of such regions has increased. So as you start taking pairs that are further apart, you see more and more of these recombined regions falling. And here, if I go only to the 8th percentile closest pair, this is sort of a 2-fold more divergence. Now, the number of, it becomes much harder to see which regions are still clonally inherited, right? So what we observe is that actually for the large majority of pairs, none of the DNA in their alignment was inherited from their closest ancestor, right? So for more, it's probably best to show it in this way. So we built a mixture model where we model the alignment as a mixture of clonally inherited regions and recombined regions. And so for each pairwise alignment, we can calculate what is the fraction of the genome clonally inherited and what is the divergence in the clonally inherited regions, what's the divergence in the recombined regions, and from that we can estimate what fraction of the differences between the pair of strains came from recombination versus came from mutations on the clonal phylogeny. And then you see that up to some distance of about 10 to the minus 3, most of the genome is clonally inherited. Then there is a region where basically now the genome gets covered by these recombined regions. And by the time you're at somewhere around 1% diverged, now all regions in the genome have been overwritten by recombination and nothing is left. And in fact, 80% of the pairs of strains that we have lie in this region where none of the DNA in the alignment stems from their clonal ancestor. Everything has been overwritten by recombination. But even for guys where there is still a substantial fraction of clonally inherited DNA, actually more than 90% of the SNPs comes from these recombined regions. Okay, so this is this pairwise analysis first indication that this recombination dominates for all but strains that are extremely close. But even for strains where still most DNA is clonally inherited, you can have that most of the SNPs come from recombination. All right, so to explain some of the other methods we applied, I first have to tell you some basic statistics about this core genome alignment. So our core genome alignment is 2.5 megabase long, but in that alignment, 90% of the positions is completely conserved. All our strains, 92 strains, have the same nucleotide. And in 11% of the positions, you find polymorphism. But if you now look within the positions that have polymorphism, 94% of these positions have only 2 alleles. Okay, so it's extremely rare that you find 3 or 4 alleles, and you can do some very basic modeling that shows that whenever you see a column where 2 nucleotides appear, this column has underwent 1 mutation in the history of the strains in the alignment. That is to say, almost all of the SNPs that we see correspond to single mutation events in the history of that position. All right, now that is actually now very helpful because that means every time you see a SNP, it gives you basically 1 bit of information about the phylogeny at that position, right? So here's a sort of a little cartoon example. So I'm looking here at a SNP where strain A, B, and C have letter A, and strain D, E, and F have letter T. And this means that whatever the phylogeny is at this position, it must have inside a branch, where on one side of the branch are strains A, B, and C, and on the other side are strains D, E, and F. And the mutation happened on this branch, separating strains A, B, and C from D, E, and F. And so in this way, every time you see a SNP on your alignment, it tells you about 1 bipartition that must occur in the tree at that position. And so now what you can do is you can look at sets of these guys. And if I say, well, let's look at X and Y, both of these are consistent with a common tree. So you can now use this pair of SNPs to say, whatever this tree is in this region, it must have both these bipartitions in them. But you can also have clashes. If you now look at positions X and Z here, you see that there is no way you can have 1 tree that has both this bipartition and that bipartition. So this tells me that somewhere between X and Z, the phylogeny must change. All right, so every time you go along the alignment and you find that the new SNP is inconsistent with previous SNPs, you know there must have been at least one recombination event. And so you can now use this to, for example, ask, as I go along the alignment, how long are stretches of mutually compatible SNPs? And how many times minimally must I change the phylogeny as I go along the alignment? All right, so if you do this, we find that stretches of 3 compatible SNPs are actually very, very short. So typically within, 4 to 8 SNPs, you will find a clash. So, and from that, we can show that the phylogeny must change more than 43,000 times along the alignment. And this is a lower bound because it's quite possible that many times you have a phylogeny change, but you simply doesn't lead to a clash in SNPs. So this is really only a lower bound on the number of times the phylogeny changes. And together with estimates that we've made from the pair analysis of the size distribution of these recombination events, you can then work out that in this alignment, a typical position in the alignment was overwritten somewhere between 100 and 1,000 times by recombination. All right. So the clonal phylogeny information has been overwritten many, many, many times in the history of the evolution of these strains. This is not unique to E. coli. So we looked at a number of other bacteria, and basically with the exception of tuberculosis, which is evolving mostly clonally or entirely clonally, is also consistent with the data that we see. All other strains show this, the fact that recombination is dominating sequence evolution. And so more than 80% of the pairs of the strains are fully recombined. Phylogeny changes every 1 to 8 SNPs. And so this is a pattern we see across a wide range of bacterial species. All right, now the other thing that we noticed, and I think is very important, is not just that recombination is so common, but that it's also highly structured. And so to look at that, I again go back to these SNPs. And I'm now going to look at a special type of SNPs. I'm going to look at SNPs where only 2 strains in the set share a nucleotide that is different from all other strains, right? So imagine I find the position in the alignment where strains X and Y have 1 nucleotide and the rest have another nucleotide. That means that at this position in the genome, X and Y must be neighbors, nearest neighbors in the phylogeny. And a mutation occurred on the branch going to the common ancestor of X and Y. Okay. So you can now play this game and take 1 strain of interest, in this case A1, and ask what are all the other strains with which strain A1 occur in such pair SNPs. And what I'm showing here is all the other strains in our collection that have pair SNPs with A1, and the thickness of the line shows how many of these pair SNPs it has. It's, in fact, the thickness is proportional to the logarithm of the number of pair SNPs. So what you see is that if there was really a clonal phylogeny, A1 could only be a nearest neighbor with 1 other strain, and so there would be only 1 link. And this is not what we see. It has pair SNPs with lots of other guys. But if recombination was totally random, it should have equal numbers of pair SNPs with everybody else. That's also not what we see. We see actually a very wide distribution with some pair SNPs occurring many times, whereas others don't. Right? So this is, for example, the picture that you get if I now overlay onto the core genome phylogeny all the pair SNPs with thickness proportional to the logarithm of the number. You see that there's a lot of exchange. There's a lot of occurrences where many different pairs of strains are nearest neighbors in the phylogeny in different places, but with very different frequencies for different pairs. If you now look at actually what is the distribution of the thicknesses of this line, what is the frequency distribution of different pair SNPs, you find that that is approximately a power law distribution. So it's a very long-tailed distribution. Most of these pair SNPs only occur once or a few times, but some of these pair SNPs occurs many thousands of times. Now, of course, you can also play this with, instead of pairs, with triplets or quartets or quintets. And so what I'm showing you here is actually that all these distributions of the occurrence of different triplets or different quartets or different dozens of SNPs are all long-tail distributions. And there is some systematic way in which the exponents of these roughly power law distributions changes as you increase the number of guys in the subset. All right, another way that you can look at the sort of the structure in these recombination between different lineages is to make what we call an entropy profile. So notice that if I have some strain S and I take some number n, let's say I take n is 4, so I'm going to look at quartets. So now let's look at all the quartets in which strain S occurs. Each of these quartets is inconsistent with each other, right? There's no, you cannot have two different quartets in which S occurs in the same tree, okay? So if I see different quartets of strains in which S occurs, each of these quartets must correspond to a different tree. And what I can now do is I can look at the frequency of all these quartets, and I can look at the entropy of this frequency distribution. So that's a measure of how diverse are the quartets in which my strain S appears along the alignment. And I can do this for each strain, and I can do this as a function of n. So I can look at pairs, triplets, quartets, and so on. And if you do that, this is what you see. So this is actually the distribution of these entropy profiles for all strains in our collection. And this is for some subset that you can see a bit better the different individual lines. And the main thing that I want you to get from this is not only that this entropy goes up, and so that there is quite some diversity in the phylogenetic structures in which each strain occurs, but also that this profile looks different for almost every strain. All right, so for almost every strain, there seem to be different statistics of the frequency with which its lineages occur in n SNPs with the lineages of all the other strains. This is just to show you that we find similar patterns across these different species that we looked at, but that if you look at the details, that both the exponents of these power law distributions, as well as the shapes of these entropy profiles, are different for each species. Okay, so these statistics encode something about the population structure, about who's exchanging DNA with whom. All right, the final thing I wanted to say is that we now think we understand the solution to this puzzle. And the solution to this puzzle is that no, the phylogeny that you converge to if you take many alignment blocks is not the clonal tree, but it reflects this complex population structure of the statistics of which lineages are exchanging DNA with which other ones. And to sort of maybe try to convince you of this, I'm showing you here that if you take an alignment of 40 human genomes from the 1,000 Human Genomes collection, and you take any large number of blocks from that alignment and build phylogenies, you also converge to a stable phylogeny. And this stable phylogeny puts together individuals that came from the same geographic ancestry, right? Europeans go together, Southeast Asians go together, and so on. And so this shows that these trees that you convert to actually reflect a sort of an average of the statistics of who is recombining with whom. All right, these recombination events, I haven't looked at all, I've only looked at the core genome, but it's important to realize that these recombination events can also change gene content. You can have a piece of DNA that has homologous endpoints to a piece of the recipient genome, but that carries in it genes that the recipient genome didn't have. And now if you do the recombination event, you've now added genes to this genome. So these recombination events also drive changes in gene content. And I just wanted to point out to you that actually this is still an order of magnitude more changes than the changes to alleles of genes that are introduced through recombination, right? So if you look now here in this plot, every dot is a pair of strains. This is the fraction of the genome that is in mismatches, and this is how many genes are different between them. And here I'm showing you what you would get if the changes introduced through SNPs were the same as the changes introduced by genes. And you see that that these changes that are introduced by gene content changes are much bigger. And we also observe, and this is just a fun observation for people that are interested in it, that the number of differences in genes, although it's highly variable, it seems to scale roughly with the logarithm of the divergence between the genomes. And I don't know why. I mean, I have some rough ideas, but there's no explanation for this yet. So with that, I come to the end. So I think what we at least believe we've learned from this is that many bacteria mostly evolve through exchange of DNA with other bacteria in their environment. This introduces new alleles of existing genes, but also changes in gene content, with the latter likely really dominating the changes to the phenotype. The exchange of DNA is not random, but there is a complex population structure, that is, which strains are more or less likely to exchange segments. And it's currently not really known what is setting this complex population structure. Is it some sort of ecology of which strains are more likely to meet? Is it maybe mostly determined by which phages infect which strains? Or is it really reflecting selection, right? So that due to functional constraints, certain recombination events are selected against, whereas others are not, and that this is really setting the changes in these observed rate. And I just wanted to say that we currently have no good models that I'm aware of that can explain the kind of statistics that we've seen in this data. So there is really a need to think of clever models with, few parameters that can sort of explain the kind of patterns that we see in this data. So with that, thanks for your attention.
Okay, I do the clapping for everybody. And thanks a lot for this fascinating talk. So please, people ask questions. You can ask them directly. We are going to record this. If you don't want to be recorded, you can just type your question in the chat, or if you have, I see already, Florence, you want to ask your question directly, or you want me to read your... So there is a question for Florence Debar. In which respect is the method you use limited to bacterial genomes?
I hadn't really thought about that, actually. But well, I mean, this whole thing with okay, so in obligate sexually reproducing organisms, we know that basically half of the genome comes from one parent, half of the genome comes from another parent every generation, right? This is still quite a bit different in the bacteria, where, I showed you that you can take a pair of strains that are at sort of divergence 4 × 10⁻⁴, and there were only, less than a handful of recombination events between them. So of course, these kind of methods of looking how the recombined regions grow as you get pairs that are further apart, this is of course not going to apply to sexual species. But sort of the method for using these clashes in the phylogeny to put an estimate of the number of recombination events in an alignment, these methods actually have been applied already for a very, very long time in alignments of sexually reproducing species. This is, which goes back to the 80s or something, right? So these kind of methods are not new in this context. This thing of these n-SNP distributions in their statistics, I think this can also be applied to genomes from sexually reproducing species to capture sort of population structure. I see no reason why you couldn't use those.
Okay, there is a question from Hector Montiel. Did you identify if the region where recombination was less common maps to some function, let's say ribosomal genes, for example?
Yeah, so, okay, so I first want to say that we don't see any regions that are excluded from recombination. All right, so the idea that there are some regions left that are not affected by this, we don't see any evidence for this. But of course, it's true that if I look at individual genes and I try to estimate how many recombination events were in one gene versus another gene, first of all, you have to realize those estimates are only lower bound. But second, the actual rate of SNPs is already different in different genes, right? So in ribosomal genes, you will see a lower density of SNPs. The SNPs that you see tend to be even more biased towards synonymous SNPs than in other genes. And So there are complicated confounding factors that it sort of would take too long now to explain, that make it hard for me to really estimate in a reasonable way the relative rates of recombination in different individual genes. But we do see differences in these rates, but it's hard to really map into a difference in recombination rate because of these confounding variables.
Okay, there is another question from Prabhav. Great talk. Is extensive transfer among bacteria suggestive of Lamarckian evolution? Can this be validated, falsified statistically?
I don't know why this would be Lamarckian, right? It's still genomic evolution. I mean, you already have Lamarckian evolution in bacteria. You have CRISPR. So, I mean, why do you need more?
There's a question by Mercedes Pascual. Any hypothesis on why the tuberculosis bacterium remains clonal?
Well, because it doesn't have an opportunity. Well, okay, so I know very little about tuberculosis, but my understanding is that this is can only reproduce in human host, and so basically the only opportunity tuberculosis would have to exchange DNA with another strain is for people that were infected with more than one strain. And so if that is rare, then one can imagine that exchange is rare. But I mean, I have to say, I haven't really studied or looked at the literature of what the current thinking is of why it's so rare in tuberculosis.
Okay, thanks. And the last question by Martin White. Did you look into patterns of recombination between non-synonymous mutations? Random recombinations between amino acid changes might be dangerous.
Yeah, this is something we're trying to look at, but all right. So let me just tell you a few facts that are relevant for thinking about this. So synonymous mutations are much more common than non-synonymous mutations. However, if you now stratify all your mutations by the frequency of the allele, right? So SNPs shared by one guy, by two guys, by three guys, by four guys, up to whatever, 40 guys. Then you find that actually non-synonymous mutations are more common in SNPs with a low frequency. So SNPs that are unique to one guy or that occur only in a pair or a triplet. And non-synonymous mutations are much rarer among, let's say, SNPs that occur among 40 guys. But it's also the case that if you have a SNP that only occurs in 1, 2, or 3 guys, it's much less likely to introduce a clash than a SNP that occurs in 40 guys. Okay, so there's a systematic bias that it's easier to detect changes in the phylogeny when you have old SNPs that are shared by lots of guys, then when you have these young SNPs that are shared by only 1 or 2 guys. And therefore, there is this systematic confounding effect of selection being more common in young SNPs, sorry, non-synonymous changing being more common in young SNPs than in old SNPs, but those are less likely to introduce clashes. And therefore, there is this systematic correlation altogether. And at that moment, I still find it hard to disentangle this from actually saying how much has selection selected against such recombination.
Okay, thanks a lot for your talk and all this good